I think it would be great for you to try and cover some of that material and practice the exercises, because they are designed to kind of go together with what we are going to be doing today. All right. And by the way, Mikhail and Cam, have you gotten the results? Have you looked at the results? Did you kind of make sense of what you found in the outputs? Okay, I'll take that as a yes. Okay, just finished. So great. So okay, great. So we'll take a look at some of the results together and try to make sense of those. All right, so today we're going to continue speaking about microbial communities, and today is kind of the second project. The second project that we'll do is related to diet and anxiety. We'll kind of briefly look at the results and discuss the different inputs and outputs, but we'll also touch on another project where it is much more clear what the differences are, but we'll kind of do that using phyloseq based on already processed data. Okay, so what I've seen is that many of you have been continuing to run some of the pipelines, and it's great to see that we are continuing to kind of utilize some of these resources. We will, after today's session, have 2 more practical exercises in R based on what we do in today's session. So for those of you that want to continue and practice what we're doing here over the weekend or for next week, you can you'll have access to that, and we'll email you those course links. So last time, let's briefly remember what we did. We discussed gut, skin, and mouth microbiome. So we saw how this project is the world's largest crowdsourced citizen science microbiome research project, where we saw a lot of data. As you remember, over 25,000 people have submitted their samples, and there's a lot of phenotypic information that we reviewed last time about these different participants. We also saw some of the important findings from this data set, from the analysis that people have done on this complete data set. And those have been published. So I don't know if you had a chance to review some of the articles that we discussed last time. There was an article on, a review article on kind of where microbial metagenomics are used in research and how that field is evolving. Has anyone looked at that article, or should I remind you where to find? Maybe we'll quickly remind everyone where to find that link. So the link was posted on the forum. Additional resources for the program. And by the way, briefly, I'll just address 2 questions that I'm aware of. Here are the 2 publications. So please take a look at those whenever you have time. I think they were worthwhile to go through. Okay, so 2 questions briefly before I continue and I forget. One question was posted by Janani, I believe, even though I forgot the name, I'm sorry, but I did see a question. And the question, okay, so JDeb, yeah, sorry, so JDeb, the question was how to find files of specific type on NCBI. So we'll take a look at that actually in tomorrow's session, or not tomorrow's, but our next session. We'll take a look in more detail on how to find specific things on NCBI, but I have been asking you to find files, so let me just briefly take a couple of minutes and look at that. So on NCBI, whenever you search for something, let's say microbiome, and you want to find those SRA links, yeah, it's not easy to kind of sort through everything, but here you have a few things that you can sort through. So you have here Illumina, and you have V4. And the thing that, whenever we talk about microbial studies, that's the predominant way of using metagenomics to study amplicon, is by using amplicon data. Obviously, if you go for the single ones, you can see samples of skin. Let's see. Okay, yeah, this is amplification of V4, even though it is single. But in some cases, that's one way that you can find those. And also, before you get to this place, you can also look for, let's see. And JDeb, are you looking specifically for 16S, the whole gene, or are you looking for, whole genome or metatranscriptomics? What exactly are you looking for? Metatranscriptomics. Well, metatranscriptomics is, very new, and I think you can just search for it like this. But if you are looking for non-amplicon, right? Non-amplicon, right, whole genome sequencing, that will provide you with a little bit more resolution into how you can study the microbial communities, but it comes with a lot of challenges. So I don't know if you want to try and go in that direction. Just based on what we've covered in this program, I think you would have to kind of familiarize yourself with a few additional tools. But I suggest looking through what we have done so far. If you can focus on some of the examples that we've done. So there are two examples. There's full 16S ribosomal RNA gene, and then there's amplicon data. And then once you kind of get a sense of what that can provide you with, you can expand that with some additional metatranscriptome data. I don't think that you will find as many. So you can see 478 compared to, hundreds of thousands. So this is something that, you can think about in terms of the variety of data that you can find. But we'll discuss this again in our next session. Now, another question was about the platform or the playground. So I wanted to clarify something about linking files. So linking files, right? Someone posted on the forum, you were trying to provide some link to a Dropbox file, and it didn't work. Let me see that specific post. Okay, there, Prerna, yeah. Okay, so you got it. So one useful way to think about this is either to use GitHub, right, that provides you a direct link to the raw file, or to use Google Drive. And there's a conversion site here once you upload your file, how to actually get the raw file that you've uploaded. So let me know if that works for you, Prerna. Okay, Divya has a question about merging reads in the pipeline in metagenomics since the DNA is double-stranded, so there will be two reads. No, paired-end reads don't have anything to do with the DNA being double-stranded. It's more the sequencing. So whenever you produce a read, right, where does that read come from? it comes from cDNA that is amplified. So you have short pieces of cDNA, and because it's DNA, you can read them from both ends. So you can read them, the same portion, read them both directions. So one from one end, one from the other end, and then you can combine them together to make sure that your sequence is accurate. So that's really the idea here. Okay. All right. So sorry for skipping over that. So let's go back and start over. We were talking about what we started doing last time, and which was discussion of the American gut data set. We looked at some of the important findings, and then we jumped into the data. And has anyone looked at the data available for the American Gut Project? Has anyone tried doing something with that? For example, in the context of what we will do next time, it's a repository of a lot of different types of samples that you can study and understand what do they tell us about healthy and what do they tell us about unhealthy or different types groups of patients or participants. All right, great. So Mikhail here has a chart. Wonderful. And can you tell us what is this for? Okay, great. Gregory found a project, and Cam, okay, producing plots. All right, so this is a stacked bar plot. Yep. And for which, okay, so I'm assuming this is for the two groups. So we have here, so we have here the different samples, and we've got here, and it's a very nice visualization. So this is really good. I don't know, have you thought of what this means and what might it tell us about the different groups that we have here? Okay, so today we'll kind of talk a little bit more and try to produce something similar. Yeah, so that's fine that you didn't divide them into groups, but it would be good to think about, what does this mean? Essentially about the samples, about the conditions, and about what we can, interpret, take away from here. But we'll talk about this here in a second. So let's remind ourselves what are the different types of samples that we have access here to. We have people that exercise rarely and people that exercise regularly. So one group reported that they never exercise, and another one says that they exercise daily. But other than that, there's also a lot of variation between them. For example, there's one person in this whole group that has their tonsils removed, and all kinds of other important or interesting anecdotal information about these participants. And what we are trying to extract from this idea is how do they compare as a group? We assume that there's going to be a lot of variability in between individuals, which we see in what we did last time and what we just saw from Mikhail's chart. But the question would be, well, how do we know what is going to be the overall conclusion from this, right? And so we'll kind of move into what we will do with the data to pipeline. So last time we briefly saw how can you produce a bar chart for just one patient or one participant to understand the composition of that participant's bacteria. And we saw firmicutes were abundant, proteobacteria and Bacteroidetes. So we saw kind of the major three participants that we've seen up until now. But what was interesting is that in the context of this whole project, each participant gets a report, right? And in that report, what do they show? They report that you compare to some other participants, just like we saw in Mikhail's chart right now. And also you have other types of kind of variability compared to other groups or between different body sites or different nationalities, etc. So now what we wanted to do is try to create a report. And this could be a report based on what you found as an interesting participant against everybody else. For example, out of the 17, maybe you could find one that stood out to you and compare them to the rest. Or you could just divide up the two groups and say, okay, so here's how the two groups compare. So since I see Mikhail has done this, but maybe some other people had trouble or maybe have not had the time to look into it, let's briefly take a look at how we can do this together. So let's go to the playground, and we'll kind of try to address some of these questions about how to do this without having running into technical issues. So please go to our code place. Yeah, one sec. Put this in chat. Okay, there we go. So if you can navigate together with me to the R playground, and please let me know once you are there. Okay. Okay, so let's wait until maybe a couple more people get there. All right. So 1, 2, 3, 4, 5. All right. Great. Excellent. So here's the link to the file where this information about the 17 samples is. So the first thing that we want to do, since this is an Excel file, is we want to get the library, open XLSX. And then we just want to load a table, right? So we'll call this OTU table. And we are going to do read XLSX, and then we are going to paste this in there. And we need the sheet number 2 is where the actual data is stored. Okay, so let's just see that we got it. OTU table. All right, success running code. Zoom in. Okay, so sorry. So did everybody load the file? Okay. Well, again, remember that these exercises are in the first lesson that is available to load and visualize basic data. So my hope is that by now, most of you can load in the data and get the right data into R. Okay, did everyone get, again, to just view what you have loaded, you can use head just to find, kind of the top few rows of your data. So if you want to use this as well, you can paste that. So let me know once you run this, do you get this success running code and you see something here in the console? All right. He's the first one that got it. Anyone else? Okay. It's because you don't have the N. It's open XLSX. You're missing the N. Okay, so it would be like that, Gregory. Open XLSX. Yeah, you gotta keep the library with lower L and without a space. All right, so the next thing that we want to do is we want to store the names of our types of OTUs by phylum, right? So if you look in this table, you can see that it's going to include the name. So let's put that into, Gregory, were you able to get it to work? So let's create a, okay. Can you just try copying and pasting this? Okay, so in the meantime, let's just create, I just did. Okay, let's create, let's store our phyla names. So we'll call this factor and out of OTU table. And we're going to take this from that first column. All right, great. Okay, so we're going to, yep, and we have the closing already. And let's just, oh, this is incorrect. Okay, so we're just gonna put here the factors from this table. So let's do head, or let's just print, or we can just put phylum. This anymore. Okay, so you can see that now we are storing all of our names in phylum. Okay, so for those of you that have, so once we store this, we can remove from our OTU table, remove that first column. And to make a bar plot, we actually have to turn this into a matrix. So we can make our first data frame and make it as matrix and OTU table, row names true. Okay, so once you do this, really all you have to do is just create a bar plot, and you can just do df1. Okay, and so here you should have a bar plot. So the whole trick of making a bar plot is really just to load in the data, make that data into a matrix. Really, just to make a bar plot, I don't even need to have this. I don't need to store these. I'm just going to use these for my colors. But if I don't want the colors, I can just use this. And this is a stacked bar plot, right? So the bar plot that we saw before that had them all stacked one next to each other, now is stacked up in one line per each sample. Now there's a lot of problems with this bar plot in terms of how we see the labels, and we don't know what these little boxes mean, but it works, right? So has anyone gotten this to work on your end? This is the code. Okay, does it work for anyone? All right, excellent. Anyone else? All right. And so Cam, let me know where in this part you had the issue. But I'm happy that, so is anyone else kind of following along? Just want to make sure that we don't go too fast. Briefly, let's introduce here a couple of changes so that we can, first of all, to make our labels, instead of going horizontal, to go vertical, which will allow us to see all of our sample names. We can do this. So just run that. Last equals 2. Okay, now you can see all of your samples, and obviously they're still cut off, but we can, first of all, let's just change also the colors. So we can make it rainbow, and we'll use phylum as our colors. Okay, so that's because it's not rainbox, right? All right, and now we have our beautiful little plot. It doesn't tell us that much, but at least we got it to display with colors, and we kind of know what these are. Really, if you look at the samples, they are different by these names. So let's not worry about right now how to modify everything in this particular group, because what we want to do now is we want to actually compare the two different groups, right? And that's going to require a little bit more kind of playing around with the data before we move in. Okay, yeah, so most of the time, the errors are going to be spelling or some kind of syntax. Okay, so first thing that you will notice is that there's just way too many colors here, right? So there's just way too many things in our table. The phyla, there's so many different ones. So first of all, we want to sum by phylum, okay? So let's briefly remove this. Well, yeah, we will add it in a little bit, but let's first of all summarize by phylum. So we can take the OTU table. One thing that we can actually use is a library called Dippler. So and another library just we will use later is called Tibble. Okay, so here we'll make a new a new data frame, which is going to be called OTU table group. And what we'll do here is we'll look at the OTU table, and we can group them by OTU. So we can use this expression like this to group by OTU. And we can also summarize all. So we're just going to make everything also go a little bit faster. So we will do, all right, great, Gregory. All right, so now we're going to just make it summarize all, and we can use a sum. So we want to just sum the table by different phyla. Okay, so let's do this, and we will also make sure that our row names are back in this OTU. Oh, what did I do? Okay, so row names, we're going to add to OTU table group. And we will make that OTU table group OTU. The reason why we're doing this is because we want to make sure that we only have the data frame. So if you do this, let's just do head, again, what did I forget? This, okay. So here, let's just do head OTU table group. Okay, let's try to run this, see what happens. Okay, so here is our new, you see the names are going to be in these, what are they called, quotation marks, and this is going to be our data. So this is 13 more variables, so 13 plus 6, right? So it looks like we're going to have a 20 different OTUs instead of the many that we had before. Before we had something like 56 levels, right? So you can see here, so what was this? This was 56 different phyla, and now we reduced it down to 20. Okay, so if we just do this, okay, so yeah, 13 more variables. Okay, let's see. So this is what I've got so far on my end. Let me know if it works for all of you, in case you're following along. All right, great. So we can continue on. So the next thing that we want to do is we want to filter out the non-zero phyla. So if the sum of all phyla is 0, we don't need that at all, right? So what we can do is we can do OTU table group 1 and add their OTU table group and have row sums. And we'll do OTU table group, and we will take, we have a total of 17 samples. So these are going to be 2 to 18. And we're going to say the ones that are more than 0. Row names true. All right. And let's also store the phylum just like we did before. So phylum, and we had our factor. Row names is misspelled. Yep. Row names. Thank you. Okay, phylum. We will take the factors from OTU table 1, OTU, as we did before. And we can also store the unique values from here. So who knows how to get unique values from phylum? How do we get the unique phylum from here? Who knows how to do that? Yep, unique, great. So unique of what? Unique of phylum. All right, now let's take and make, okay, so because our OTU table group has the names of the, the names of our phyla in column 1, and then all of the rest go on after that. Okay, so then we want to create our bar plot, right? So let's make a bar plot. So what are we going to bar plot here? What do you think we are going to use as our data for bar plot? What would I use here? Where's the data frame that I need to use? What's it called? Yep, OTU table group 1. Yeah, so it's a warning, right? Gregory, this is a warning, it's not an error. Okay. All right, great. So yeah, let's put here OTU table. Let me just copy this. So this is what we want to use to bar plot. So let's just run this. Okay. Height must be a vector or a matrix. So here, right, what we need to do is we need to make sure that all of the information before we run this, right, we need to convert it to a table. So we're going to use OTU table 1. Oh, hold on a second. No, we need to make sure that we have named all of our, yeah, so we need to make so we can make a matrix for this, or we can make a table, first of all, for our OTU table, right? So if we take OTU table, let's just try this real quick. OTU table. Height must be, yeah. So here, what the problem is that every time that we take these, we're going to run into this problem with the column or the row names being in a column. So for example, if we want to take OTU table, what we need to do is we need to convert it to a table, removing, so let's do this, OTU table. 1, and that's going to be table, OTU, table, and then just take the first column, right? So this right here will give us any sample that we want, right? So if we just do a table of 1, this will give us 1 sample. If I want to do, let's say, number 17, right, I can get number 17. So, and so forth. But what I need to keep in mind is that I need to store my information about the phyla. So this is for 1 sample, we already did this. So let's now take for all samples. For all samples, instead of a table, we need a matrix. So let's call this a data frame, and we'll just call this as matrix. And we need to take OTU table group 1. And we need to remove the first column, which is 1. And row names equals true. And now we can bar plot our OTU table group 1, or DF, right? Because we changed it. Okay, what happened here? OTU table group 1. Oh, I need to remove 1 instead of just keeping column 1. All right, so as you can see, this is much less cluttered now because we only have 20 instead of 56. Great, thank you, Michael. All right, so this is now a comparison between our samples. And here we can again say last equals 2, and that will give us the names of our samples. Right, now we can kind of see them. And we can also add color, which would be phylum 1. We can say rainbow, phylum 1. Here, again, rainbows. Missing a bracket at the end. Thank you. All right. Okay, so the problem is here, we just need to get the length of phylum 1. Or actually, we need to have phylum is what we need. Let's try that. All right, but it's all red. So no, we need the length. Thing here. Library open, okay. Some by phylum, what is it not liking? So phylum 1. Missing a bracket at the end. Oh. Okay, so Gregory, all right, great, there we go. Gregory, argument row name's true. All right. So is everyone getting this? Yeah. So let me paste this code here. Okay, for Gregory, OTU table, you have another O there in OTU table group 1. You've got an extra O. Okay, so let's quickly get to making a group. Okay, we won't worry about making this plot look nicer. Let's just make a plot for the group. So we'll just prepare 2 groups. How do we do that? So first of all, we need to create those 2 groups. So let's make one group by group rarely, the exercise rarely. And we'll say as matrix, and we will take row means, right? Because we want to take the mean of all of our phyla between samples, and we'll take OTU table group 1. Now, if you look at this, you know that the first 7 are actually the ones that rarely exercise, and the last 10 are the ones that exercise regularly. So here we will say we need to take only 2 to 8. And the other group is going to be called by group regularly. And here we'll have as matrix row means OTU table group 1. And here we'll have 9 to 18. Okay, so now it becomes a simple problem. We just need to combine them together, right? So let's just do groups, and we will say cbind, that binds them together. And we need to say by group rarely, and by group regularly. And let's just add those names also. So we'll just say OTU, or sorry, call names. We'll just add call names to OTU groups. And they will be rarely and regularly. And the row names are going to be, for this, are going to be phylum 1. And now we can plot it. So again, we can do bar plot. But here, instead of this, we will use OTU groups. Okay, so did anyone get this? Again, just so you can try it, and this will be as a lesson / tutorial also. But this is a very simple code that allows us to group our information by mean values between the groups. and try to compare them to each other. So obviously here, we don't know what all the information is, and we need to know what the colors are. But even if we add the colors, there's quite a number of different colors here, right? There's all kinds of colors here up top. So a useful way to do this is to instead print a nice table. Now, when you want to produce a table output, you can make an output in the console, for example, right? But that doesn't look very good. And we want to create a report. And so when you want to create a report is you want to maybe create a PDF. So a useful library for this is called Grid Extra. It's actually useful also for a lot of other things. But in this case, we'll just use it to make a simple table. And when you want to print a PDF, you can just type PDF and open up a PDF file, for example, groups compare, and then add grid table and put our OTU groups in here. And to finish making that file, you just print dev.off. Again, sorry, I just don't want to run out of time, so we're going to move a little bit quicker. Okay, so here is our result, and we can see here right away, for example, actinobacteria is more for those that exercise rarely, and Bacteroidetes is less than those that exercise regularly. It's less for rarely and more for regularly. Those are the things that kind of stand out right away. Here, firmicutes, less for rarely, more for regularly, right? And here you can see, for example, tenericutes, it's 2 versus 143. So what are these tenericutes? Again, intestinal microbiome in the liver. So we can now study specific representatives that we think are more informative, right? So here I can see almost twice as much proteobacteria than here, right? Now, obviously, this is not a very statistically rigorous analysis because we're not counting the variation within each group, right? This is just something that we know about the group as a whole. But if we look in here, right, this is kind of the first 7, 1, 2, 3, 4, 5, 6, 7. These have a lot of variability. You can see this guy right here and this guy right here. So you do have a lot of variability within both groups. Now, what is the statistical test that you know of that allows us to account for variability and compare the means between two groups? No. Yep, just a statistical test. Yep. Well, ANOVA is kind of going very far with it, but yes, it's what other names do you know? T-test, excellent. Yeah, so T-test, that's exactly what T-test does. T-test compares the means between two groups and incorporates variability, divides that by variability in each group. And what is the output of T-test? What would be the output of T-test? Who knows what does T-test produce? Right, a p-value. Great. Yeah, it produces the t-distribution, and you can calculate the p-value from the t-distribution, which is going to be how significant, right, is a difference, how statistically significant. So the way you do this is t.test for the two groups, which is by group rarely. Well, sorry, before, yeah. Well, okay, you know what? We need to get on to phylostick, but when you have, so we already calculated the means here, so we can't use by group rarely, but you need to just take this, and you write t.test between this and this. Okay, so here we have our output. Okay, and so we actually probably need to do this, this is probably just for the first row, so we got to do it for all of the rows. So I assume if you do something like, let's see, where was our output? Let's say, let's say this. So this is 1, 2, 3, 4, 5. Let's take row 5. Row 5. Okay, so you see that the p-value is not good, because there is a lot of variability, so we could just be getting these differences by chance. Okay, so just wanted to briefly introduce you to how we can use some of these things and study differences between groups, making sure that we're also thinking about how this is going to be useful for biological interpretation, right? So it's nice to kind of have an output like this to see what are some of the differences, but we still have to check for variability, and that does play a big role. So before we run off, let me just, because I'm just interested now, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Let's try 15. What will happen? Did anyone have any questions about this code? Still not good. Did anyone have any questions about this? No. 15? Yeah, it's just row 15. T-test is done for each one of our rows, right? Because each one of our rows is an observation. So here you have, remember that we have 7 different observations here and 10 observations here. So we're comparing, this is the mean, but then we're also accounting for the variability within this group and variability within this group. So when I just do t-test, it's going to take the first row. But then if I specify the row, that would be row 15, because I was interested in checking this one, I think. Right. Okay. Any other questions about what we did here? Okay, and again, we will have this. As something. Oh, I don't know why I can't paste. Strange. Okay, don't know what happened, but can't paste. But I will make sure that we save this for everyone to practice if you would like to. Okay, so since you kind of know this, multi-column table for all species. What do you mean by multi-column table? Oh, okay, if you want to, instead of looking at, oh yes, you can, using a for function, you can definitely do that. You would have to write their name, and then create a table. So it will take a little bit of time, but I really want to, make sure that we have enough time to talk about PhyloSeq and the data to pipeline, because a lot of this is actually built in to PhyloSeq. So I just want to get to there now that you kind of get the idea of how to do that. Yeah, let's move on. All right. And Cam, was that okay? Yeah, you just pasted. Thank you. Okay, so let's talk about this second part. So we did this. Okay, so the project that we want to talk about, and actually I had two projects in mind today, but maybe we won't have time for doing both of them, but let's see if we can make some progress with this. So I know that not all of you have tried this, but I wanted to try and do this together. Let me briefly remind you what the project was and what the data was. So this is an example of a data to pipeline, which works on the read, denoising, and rarefication of taxonomy. So what do these things mean? It's kind of what we did now manually, but it does it in a more sophisticated way. So remember that when we look at the output from from this pipeline, it takes reads, make sure that those reads have accurate sequences. In other words, those reads do not contain any kinds of contamination in terms of the sequence itself. It's also denoised because they are merged, the pairs are merged. And then there's a specific emphasis on the search for chimeras. Chimeras are reads that contain a piece of one read and a piece of another read. And that might confuse our OTU assignment. So once we get to the assignment of operational taxonomic units, we want to what's called rarefy. In other words, make sure that a lot of this information here is going to include kind of noisy data, and that data might not be useful for our analysis. So it's going to try and reduce the number of phyla, if we choose phyla, for our downstream analysis. And then, you now know what these basically mean, what we just produced together. It just produces a plot by abundance of each phyla, and also looks at how these phyla are different within each sample. So when I look at a sample like this, as opposed to looking at a sample like this, how do I compare this sample versus that sample? I need some kind of a metric. So how can I develop a metric that represents, for example, the relationship between Bacteroidetes and firmicutes, right? As opposed to firmicutes versus Microbiome. So those distances, those phylogenetic distances, could be different. And because they are different, I can analyze the richness, right? Richness means how many representatives from a diverse population are present in a given sample. And also, I can do so by group. And obviously, I can also count how many species, as well as how divergent they are from each other. And to do that, what this analysis, this PhyloSeq kind of downstream analysis does is, first of all, it looks at distances, phylogenetic distances, using ape, which is just a way to construct a phylogenetic tree, and converts that into a statistical measurement that explains to us how this represents the data from the standpoint of alpha and beta diversity. So alpha diversity and beta diversity. And what we want to try and do is try to reproduce this whole process together. First of all, running the pipeline together, and then taking phyloseq portion of it and running that in R. And hopefully we'll have enough time to do that. So what we need to run this pipeline are two files. The first file are the sequences. These sequences could be from NCBI, where you found the SRA run table, or they could be already stored on the system. So here, for example, you notice that each one of the samples has two files. Why is that, do you think? Why do we have two files for each sample? Right, exactly. So every time that we want to make sure that we have good data, that's the way to check, is that it's going to have _1 and 2. The other thing that we need is this description of what are the names. So you can see that BKF01 is BKF01, right? So that's just the name of that sample and the library name. Now this right here could change, and that's why I have another example for you to try out. So let's take a look. I have here the actual links to these files. So they were also in the. Are these files downloaded in the same folder? So I'm not sure what you mean by that, but these are the links that I just gave you. Those are the links to these files. So if you just click on the link, you'll see them. It will just download to your computer. Yeah, okay, so this one shows you where the data is stored. And you can also see the second file. Oh, sorry. So yeah, so this is, these are some more samples for us to use. Okay, so how would I, for example, take the data that was originally given in Metagenomics 2? So let me show you that. Okay, if you go to the demo pipeline, okay, sorry, I'll show you where to download all of the files. So if you could please go with me to server.t-bio.info. Okay, let me know once you are here. All right, excellent. Anyone else? Just one more person. Okay, great. So you go to Amplicon 16S and just click on the demo. And this is what we will reproduce here together. So here you have your input files. And there are two links that we need. We need the SVL. So you click on this icon right here, and this reference table or supplementary table. So what are they going to look like? The links are going to look like this, right? So we had that on the screen, and the supplementary table is just the name of the sample and the library. Did everyone get these files? Yeah? Did everyone get these files? So if you got the files, maybe just put in yes, so I can see. All right. Okay, so please let me know if you haven't found the files or you're having trouble finding them. Now, instead of doing the demo, let's go into the actual pipeline, which is right here. So here now we can upload files. Okay? If you're here, also please let me know. What are the settings that we have here? First of all, these are FASTQ files, right? So we can go ahead and change that. The type of database, you can actually upload your own database. We're not going to do that. We're just going to select the Silva database here. And these are pair end files. So now we can click on File Upload. The first file that we need to upload are the SVL links. Now there's a number of different options here. These files are not that big, so if you happen to download some files onto your computer and have them stored, you would upload them from Upload Files. You can also use files from link, or you can use URL bulk upload. So if you click on URL bulk upload, we are going to use that SVL link. So that was in my downloads, and that was the SVL link. Okay. And then next you will see this button, supplementary text file. Did everyone get to this step? All right, so let's go to supplementary. Okay, that means you're in the wrong place. You probably tried using it from here, but you need to add from down below here. It should say, right, let me do this again. FastQ, select Silva. And then you don't select this one. Okay, so if you could just select URL bulk upload, because we're referencing a number of different files together. And that's where I can use the SVL. So did that work for you, Gregory? All right. Is anyone else having a tough time following this? I want to make sure that we don't lose anyone in the way. Okay, so the next one that we want to upload is the supplementary text file. By the way, what we'll go through next time is we'll talk about how to upload files directly from NCBI using that SRA run table. Okay, but right now let's click on supplementary text file. And here you can load the supplementary table. And remember this name right here, library name. So library with a capital L and capital N. That is what you need to remember. And click on, okay. So now we can start our pipeline. Ignore this. And we'll go to filtering. So what are we setting here? These are the lengths of our reads, and this information is going to be in the SRA table. So in the SRA table, you will find typically the read length, which is going to be called something like spot length. And also, how many errors are you allowing? And you can play around with these parameters. You know, some of these are going to be shorter, some of these are going to be longer, so you can play around with these parameters based on your data. Here we don't have any options. We merge the pairs. Here again, remember when we're merging pairs, we're looking at overlap between the reads. This is a fairly flexible setting, 12, and this is a fairly stringent mismatch overlap. So you can also change these. Here you don't have any settings, or here, or assigning taxon is just going to use it based on the database. But now we'll have some settings here. So first of all, APE stands for Analysis of Phylogenetics and Evolution. So why are we using APE? So what are we trying to understand the phylogeny of? What do you think we're trying to analyze here? Yeah, relatedness of what? What's related to what here? Yep, OTUs, exactly. So OTUs, these operational taxonomic units, exactly, they could be related either far apart by common ancestry or close together by common ancestry. So that's what we have here. There's a couple of different methods for building that phylogenetic tree, and neighbor training is NJ, or BioNJ is just a different method of doing this. And then this is what we also can use to visualize all of these different types of relationships. So here, again, remember that typically we're going to use phylum, which is not a very detailed view. So obviously you can try other ones and you'll run into some NAs, but still phylum provides you with a good overview. The mean abundance, we can leave that for now. Color is where we can use library name. So that will color the samples by the name of the library. And obviously you can also use library name here, or if we had two groups, right? So for example, if in our data set we had library name and then we had, for example, color or group, right? And then here had, 1 and 2, it could also be used here. And here you have a number of different ordination methods. So NMDS is just one of them. Principal coordinate analysis is another one. There's also just simple MDS and all kinds of other ones that you can select from here. And then end. So data 2, chow versus high-fat diet, fecal. And now you can run the pipeline. So let's now take a look at what this might mean. Unfortunately, we don't have too much time left. Okay, but now you can see that this is running. Now, in preparation for today's session, I wanted to introduce you to a couple of other examples. First of all, I guess we need to think about what would be the output from the pipeline. And so the pipeline is going to take a little bit of time, but not too much, because the files are fairly small. But as a result, as you remember, what we will see is a bar plot, and we will be able to analyze richness of species and the diversity of species, and see that the samples are indeed different, and primarily different by the group, but also we have some outliers, and we can ask ourselves some questions about what do these outliers mean in the context of what we have here. So this is, by the way, a different number of samples that I wanted to give you to practice what we did today. So what I gave you earlier, the links to the links on GitHub are links to files from the same project, but also comparing fecal and cecal samples. Yeah. So just in the same way that we are, we did the pipeline today, if you can try filing these files, and here just, you can also refer to the bio project where this information came from. So this is something that I'll post on the forum. Okay, so how do we do this with a data set from NCBI? So let's say you found some data set on NCBI. Okay, I am interested in the microbiome of the skin, and the typical skin condition is acne. So let's find here an example data set that we can analyze. Okay. So if I find a number of bioprojects here, you can see that some of them will include many samples. And obviously, it is not always a requirement to take all of your samples. So here, for example, you have 82 samples. You know, previously in this data 2 pipeline, we only analyzed 6 samples, and it's still going to take some time. So 82 is definitely going to take a longer time. But once you find this data set, you can go to, you can go to, there we go. The SRA. I don't know why this happens, probably because I need to clear this. Okay, there we go. And from this place, you have to download the metadata to start selecting the specific samples that you would like to compare. So remember that this is in CSV format. I just need to convert it here. And once you do, you can identify samples that you think might be interesting for a comparison, right? Our goal is to compare here. So if you go and you see here, there are several male and female patients. They have different disease type. So acne, some of them do have acne, some of them are controls, they don't have acne. And so we can make a comparison between acne and non-acne skin. So once I have done that, let's say I'll take 10 of each, or let's say I'll take even 1, 2, 5 of each, right? So for example, I'll take this first one. And I'll take these 3. Let's say these 2. If I save this, I also need that additional table. And that table is going to tell me what is this in terms of study disease. So I'm going to take another sheet here. Let's do that. And just take the run. say here control. So this is going to be supplementary table. All right, so now I have the supplementary table, which the name is study disease. And I have the acne metadata, which has all of this information. So now that I have this, I can go back and run this analysis on the platform. Again, Gregory, I will get to that. Definitely put it on the forum, and I can even email you if you'd like the data set and the specific publication about that data set. Okay, I just want to get to this place where we want to upload the file. So again, we have our data set. It's pair end, FASTQ. We're going to use Silva, file upload. And now instead of going to URL bulk upload, we need to go to NCBI run table upload. So here you add the files, and here we add the metadata. Supplementary text file. We add the subtable. And remember, when we're building our pipeline, we're going to be doing the same thing, but we're going to be using our control data, the group data, for coloring. Okay, so here, again, we'll use phylum. And for color, we will use study disease. Okay, we're already over time. Unfortunately, I did want to show you how to run the phyloseq methods in R, but instead of taking up the time, maybe we'll have some time to do it in our next session, or we will just send you a direct link to the lesson on the code OmicsLogic.com, so you can explore that. And we'll try this with a couple of different data sets that we can look at, including this acne data set, and the acne and the other one. Sorry, what is the question about the control studies? Can you clarify? The control studies, I mean, I was just referring to, yeah, so I was just referring that here they have acne and non-acne. So this is a case control study. A case control study means that they had people that are of the same sex, and you can see here that subject is affected, yes or no. So it's a case control study. That's what I meant to say. Okay, so any other questions about what we did here? I hope it made sense. In our next session, we'll get to the interpretation of some of these differences and see how to do this in R. And we will also speak about how to build your project. In other words, how to actually go through the NCBI data, find an interesting data set, prepare all of this stuff again, and then also how to think through your analysis to identify what might be the outcome. So based on what you think is the outcome, you might want to be looking for specific things in the metadata when you're preparing for the project. Again, thank you for joining today. We will see you on Monday. and please continue, try to go through Metagenomics 2, submit your project proposal. I know that we already have a few, and we're looking through them, and we'll start getting back to you about those as well. All right, great. Thank you all of you, and have a great weekend.